Hi everyone, my name is Yasha Nazir, and today I'll be talking about Gibson cloning. Um, what I'd like to cover in this video is that what exactly this Gibson cloning is, its procedure, and how to design Gibson Primer using Snapgene, and then finally, which Gibson mixture is recommended to be used in the experiments. So first, what exactly is Gibson cloning? Um, Unlike conventional cloning method in which you can clone only one gene at a time, in Gibson cloning you can clone multiple fragments to the same vector in just one tube at the same time, which is fantastic because also you don't need to have restriction digestion sites that we normally look for when we have to clone. So it's literally magical cloning method and much more advanced. So a little bit of background to it. In 2009, Dr. Daniel Gibson and his colleagues at J. Craig Venter Institute developed this technique. And a few benefits is that, as I mentioned earlier, in just one tube, you have several reactions that take place, such as exonuclease activity, polymerization, and ligation, all those. And multiple fragments can be cloned into the vector at the same time. You don't need any restriction sites at all. So this is kind of like a diagram that would show how, let's say, if this black DNA fragment is our fragment, and this green DNA is the linear vector. Now what happens is that we want to clone this black fragment into the green. So they're just showing you one side, and imagine it occurs to the other side, like where this part, this 5' end based um, side of this green would be ligated to the 3' end based side of the black to make circularized DNA. So what we normally do is, if you see that this 3' end of the green, the linear DNA, this entire part is copied and attached to the um, fragment, um, and this is done using the primers that are designed, and PCR is basically done. And then this way we get to have um, the exonuclease activity, where exonuclease enzyme would chew only towards uh, from 5' to the 3'. So, so if 5' was here first, now it is here. So because we have two DNA, so 5' to 3', 5' to 3', exonuclease activity takes place. And then now these two single-stranded DNA are complementary to each other. So they would join together, and then we have these gaps which are filled by DNA polymerase, and these gaps are once closed, then ligase would seal these nicks. So imagining that towards the second end, the same thing would happen, and moving to how the primers actually look like is, so if we have actually 4 primers instead of 2 primers that are normally used in the conventional cloning. So there are 2 primers for the insert, so forward insert, reverse insert, and then there are two primers for the uh, vector, which includes forward vector and then reverse vector. So you can notice that half of the part of the primer or the sequence is coming from the backbone, the vector, and half of it is coming from the insert. Same is the case with all of these, where they are sharing half of the sequence from the vector and half of the sequence from the insert. So overall, the protocol is such that you amplify the fragment separately and vector backbone separately in two different tubes in a PCR machine. And once you have the DNA amplified using those specific primers that we talked about, so if you are trying to amplify the fragment, then you would use um, this primer forward and this primer reverse. And if you're trying to, in another tube, you would have this forward primer for the vector and this reverse primer for the vector. But you know that this part, half of it is coming from the um, gene of interest. So when you amplify fragments and vectors separately in the tube, you would run the gel and see whether you get the specific sizes based fragments and specific sizes based linear vector. So let's say your gene of interest is 1 kb and your vector is 3 kb, then you would see a band at 3 kb for the vector and 1 kb for the fragment. Then, now that if you verify using agarose gel that you have enough of the fragment and enough of the vector, you would continue to an isothermal reaction where you use the Gibson mixture and mix both the DNA along with Gibson mixture. And here you just have to incubate at 50 degrees for 1 hour. It's just one single tube which has all those three reactions that we talked about, including exonuclease, polymerase, and ligase. And finally, you run another agarose gel to check if you get the band that would now have 4 kb of the product. So this Gibson mixture, I would like to show which one we normally order for our lab. So this is the Gibson Assembly Master Mixture by NEB. So you do the calculations based on how many fragments of DNA you have, and you have the positive control as well that comes along with the kit if you want. And yeah, you do the calculations, how much of the fragment in picomole are you going to add, and how much of the vector you would add. And then finally, moving to the primer designing using Snapgene. So what I have here is the PASC IBA5C. It's a vector. And then I would want to add the gene into the multiple cloning site. So as you know that a specific vector, any vector, has important sites to it, including multiple cloning site, origin of replication, and any resistance-based genes. So let's start with the primer designing for Gibson. What you would do is very, very simple. You would go to Actions, and then Gibson assembly, and then insert fragment. So you have this whole choice whether you want to add one fragment or 10 fragments, it's up to you. So I will be adding one fragment only. So now another window opens where you have already selected the vector. So you can click on the multiple cloning site. So this is the site where I want to add my gene of interest. And now, because I have selected the sequence, you can also view the sequence from here. You can get to see that in the multiple cloning site, how much of GC content I have, or how long this multiple cloning site is, which in this case is 97 base pairs. Then I would move to the fragment. Now, as you know, I haven't selected the fragment yet. So you need to select a file which has your gene of interest. So let's say this is APOPEC3G is the gene that I want to be cloned into that vector that I showed you earlier. So what I would do is, now because I have selected the sequence, I can even view it in the sequence file right here. It has actually two domains to it, C-terminal domain and then this orange one, N-terminal domain. So I want the entire gene. So now that this has been selected, I would go to the product. So this is how it would look like. So this is the A3G gene that I selected, and then this is the PASC IBA5 vector. So I would go to the choose overlapping PCR primers. So it gives you a TM for PCR primers, which looks good, it's 60. And then I would go to choose primers. So now that you can see, I can view my primers that have been designed. So this is just a primer that I had on my own, so just ignore this one. So now you can see that it shows me fragment forward, fragment reverse, vector forward. So these are the 4 primers that I would be needing. And in one tube, I would be adding fragment forward, fragment reverse, along with template that will be the plasmid that has my fragment in it. And in another tube, I would run a PCR using vector reverse primer, vector forward primer, along with the vector PASC IBA5. See. So then it tells you that this it's ready for Gibson assembly, and your final product would be 4072 base pairs. What you can do is you can simply rename this PASC A3G, because I'm adding A3G gene to PASC, so I can name that. I have the 2 TMs, and it's telling me that everything's working perfectly fine. So now I can click assemble. Now this is a newly generated plasmid that you can see in the multiple cloning site. Now I have this gene of interest, and the rest of this is my same vector backbone. So yeah, that's how you design the Gibson primers. So I can basically go to IDT and order the primers that were listed, and then I'll be good to go. Thank you so much everyone for your attention.